Thank you to Dan for asking me to give a talk when I came here. It's really a privilege to be with you guys here. My name is Rachel Hilmer. I am officially now a biostatistician at what has now become Koch Biological Solutions. My background is in computational biology, so my understanding of statistics mostly came from microarray and RNA-seq analysis, and now I'm moving into field trial analysis, and it's really cool, and I'm really glad I'm in agriculture, but there's a lot to learn. So yes, off and away. So I chose this picture because this field has been super, super carefully designed, how this how a tractor was going to go along this field and where the plants were going to be put, with nice borders here to correct for the gradient and everything like this. And the same level of care really needs to be taken with the statistics as well as with how we design the experiment. So that's why I chose the picture. Careful design is a beautiful thing. Okay, so just up front, I have worked with a lot of field trial data at Koch Biological Solutions, but the data that I'll show you in this presentation are generated. So they do represent the means and the variances that I've seen, but this is not an actual microbial treatment or biological treatment, just so you know. Okay. We have seen gears before today, and there's been a theme of, oh, what happened in statistics? Did statistics fail? Are p-values no longer valid? And I would say that statistics and experimentation are like gears, and if they're properly interlocked, then the statistics works. But if the assumptions behind your tests fail, then statistics will fail, and it's not because statistics is inherently bad, it's because your assumptions fail. So if instead of a bunch of gears here, this became smooth, this would be turning and it would mean nothing. So sometimes that happens to us in statistics. Okay, so I'm going to talk to you guys today about residuals. And one way that you can look into your data and see, did I violate assumptions and are my conclusions no longer valid, is by looking at residuals after you fit a model. So what are residuals? Well, so some of the questions that you can ask and figure out whether you've violated assumptions are, is the data normally distributed? So if it's not normally distributed, there will be clues in the residuals that will help you figure that out. Is the data heteroscedastic? Okay, so I put this here because it's a nasty big word that makes sense to statisticians, and often in statistics there are big complicated words that actually mean very simple things. So please don't be put off when you go into the statistics literature by big complicated words. This is really simple. It just means, is the variance independent of the mean? Do you have really big variance when you get high values of yield and really low variance when you have low values of yield? That'll produce problems. So very simple. We'll go into that. Are there strong outliers? We've heard about outliers. You can see them in the residuals. And are there additional experimental factors that you didn't think of that are influencing your response that you need to take care of? Sometimes you can see those in the residuals too. So residuals are your friend. And I will also briefly talk about a power analysis. So the residuals tell you what if the next experiment is like the previous one, which is often true, not always. The residuals help you figure out the sample size that you need to detect stuff. Okay, so what are residuals? In a really simple-minded way, imagine that you have a really simple experiment with an input and a response, and you collected some data. And so you might look at this data and say, I think I should fit a line to this data. And that would be a nice interpretive model. So the residuals are the leftover variation after you explain for the obvious effects. So here, this is the line fit that I show you. The residuals are these red lines here. So that's the inherent variation, the noise that's left over after you accounted for effects that you know about. Okay, and so if I have these lines here and I plot them, so instead of response versus input, I'm plotting residual now versus input. This is the leftover noise. And so I'm going to introduce a few straightforward things. So one thing is that they're nice. These are good residuals. They're nicely balanced around 0, positive and negative. And they're pretty flat. You don't see like really high residuals here and really low residuals here. So this is like good residual behavior. OK, right, I have this in my hand. Okay, so that's a really simplified example. Let's go to something that might be more relevant and more interesting. So imagine that you have a randomized complete block design, because you're a very good agronomist, and you designed this with a single gradient to take care of something that you know happens in this field that you've used before. You've got nice loamy soil over here, and you have clay over here. You want to correct for this. I'm not saying that this is the best design at all. This is just a hypothetical example. Okay. So, I'm sorry?
What are the colors?
Oh, thank you so much for stopping me and helping me explain this. So we have 10 different treatments here. So a control and 9 different treatments. So at Mental, we're screening for different biologicals that are active. So we have a large number of treatments that we have to incorporate into our design. But you guys have that too. Okay, so, and if you wanted to, then you could plot yield by treatment. So here's the yield data as a function of treatment, different yield in different colors. Okay, all good so far. Then I could fit a mixed effects, or I'm sorry, just a regular linear regression model to this data, and with fixed effects. So I would give a different intercept to every single treatment, estimating the mean. And if I did that, the residuals would look like this. So as you can see, I fit a different mean to each treatment. So that's why the dots stack up vertically on top of each other, because I fit a single value. And then the leftover variation in each one, that lets you know how much variation you have left after you estimated the effect that you care about. So I told you up front, hey, there's a soil gradient, and we're going to correct for it, and we have a nice design that will allow us to correct for that. But what I'm going to walk through is, what if you didn't know about that? How much would you lose, and how would that affect your power? And but I told you that it's there, so I gave you the answer in some ways. So if instead of plotting in color the treatment associated with a particular point, what if you plot the replicate? So as you can see here, it's no longer nicely balanced around 0. So we're consistently underestimating the effect in the replicate 6, which is the block 6, and then we're consistently overestimating the effect in replicate 1. So, I'm sorry, other way around. The residuals are high, so we're consistently underestimating, and vice versa. Okay, so when you look at this then, it suggests that you're missing some factor here, because you see a pattern, a variable that's not evenly distributed around 0 inside of your residuals. So there are other ways to explore your data and find this, but this is just a simple graphical explanation. Okay, so let's fit a model then properly, having fixed effects for the treatment and random effect for our blocks. And now you can see when I plot color, not only is the variance a lot smaller, the different colors are evenly balanced around 0, so we don't have systematic effects. So what's the problem? In the first experiment, you would not have had any artifacts that were just a bad design, but you would have had so much variance that you can't really say anything. So how bad is it when you aren't able to properly know what variables are influencing your system? To do that, we do a power analysis. So there are some ingredients that go into a power analysis. If you have these ingredients, you can figure out sample size that you need in order to figure out how many samples you need. Okay, so you need the effect size that you care about. You need how much you can tolerate a false positive, how much you can tolerate a false negative, and then this is the key that comes from the residuals. How much variation is left over after you account for the facts that you know about. So let's do that with these 2 different data sets, like this. So this is a visualization here, and there's several variables in here, so I'll walk through this just a little bit. So I've plotted effect size as percent change. So I didn't make the calculations as percent change, but I've plotted it as percent change just because people like to look at percent change to understand effect size. So this is effect size plotted here and here. This is when I include the replicate effect, and this is when I don't. This is sample size treatment. This is the number of samples that I will need in order to get the p-value that I want. You can't really read this. This is 0 to 500. I realize that's insane. You wouldn't do that in a field trial, but this is for purpose of explanation. And then you have to choose the power that you want. So this is how much you can tolerate false negatives. You didn't see it because you just didn't have a big enough sample size. And then you have to choose a p-value. So I chose 0.01. Now, that might seem ridiculously small, but it's really important to do a major error that I've seen in our screening pipeline is not correcting for multiple hypothesis testing. So if you test many, many hypotheses, you need to correct for the fact that you just tested a bunch of hypotheses. So if I make 10 hypotheses and I set a p-value at 0.1, which would be a standard thing, 0.1 means 1 out of 10, it's going to happen randomly. So if you test 10 things and you see one thing that shows up randomly, well, that might have just been random chance. So I have an adjusted p-value that should come out to about 0.1, depending on how it gets adjusted. There's different methods. We can go into that later. The more you test, there are different methods. But OK, so I have all of these things. And you can see that if I use this replicate effect, the axis is so small you can't quite see it. At a reasonably decent power of 0.85, I can have a sample size of 6. And I can call the significant effect at 20%, which for this experiment was good enough. But if I didn't estimate that effect, the effect is dramatic on how bad the sample size is. So being able to know what are those factors in the field that are influencing what we care about and being able to correct for them is huge. So this business with the Latin square design of having that other dimension where you can correct for things even if you don't know it, it can have a huge effect on your ability to call significance. OK. And then one other thing that the residuals can tell you are, so that's a good use of residuals. What if your model's wrong and you have some assumptions or some conclusions that you've reached and they're not valid? How would you know that? So that's what I'm going to talk about next. So there's an assumption when you fit a model in R or in ARM or whatever that the variance is independent of the mean. So your residuals should look like a tube. But sometimes they look like a gramophone or like a funnel. And I'll explain why that can do bad things to you. So imagine that instead our data looked like this. So this was our yield data as a function of treatment. Can you guys see that there's little dotted lines plotted here? No. OK, we're going to go like this. There are dotted lines plotted like this and like this. You can kind of see that there's a little bit of a funnel shape here. Can you guys see that? OK, good. That didn't show up. So what does this mean? It means that at higher values, there's just larger noise. And then at lower values, there's less variability. Well, why is that bad? Well, if you have higher variability at large values, it means that when you measure a large value, there's more variability in it. And you can get an outlier that's high. And it's not unexpectedly high, but it looks unexpectedly high because what you're calling normal is based on something that's in the middle here. So you can get outliers in one direction. OK, so how does that show up? Here's 0. And you can see that there's more stuff way up here than there are down here. So notice that this is not balanced on the y-axis. This is negative 25, 25, and 50. OK, so you can get outliers in one direction. And you can call things significant, and it's not actually significant. You have a few outliers driving the effect because you didn't have the right, you violated the assumptions of the statistics. So what do you do in this case? Well, maybe you were using a wrong metric, and you can go back to the start, which might be good. If you have enough data, sometimes, if it's all you have, you can consider transforming the data. So there's two ways that you could look at this. Maybe you're more familiar with looking at a QQ norm plot. So here you can see that these outliers up here are pulling really far away from this line, which represents a normal distribution. So you have outliers in one direction. OK. So perhaps not so much with this data, but I've seen cases where log transformation is really, really necessary in the data, that you'll have crazy outliers, and if you don't transform it, you will get really spurious conclusions. And maybe there are better ways to actually get into the model and not have to use transformations. But biomass data, sometimes one of them. Disease severity studies where infection severity is being scored, especially if that's colony-forming units that you're measuring, which is continuous, and also with water use efficiency, which is a ratio. I've seen really bad behavior, and so you have to go back and say, okay, what do we do with the fact that we totally violated our assumptions? So I'm not saying just go transform your data if you see residuals like that, but look into it and make sure that the assumptions that you are making are decent. And not that necessarily you have to do the statistics, but these assumptions are really simple. So the calculations behind this is complicated, but going in and asking, hey, did we violate simple assumptions in statistics? It can be done and should be routinely done, and if it's not, you can call things as significant that aren't. So those are just my take-home messages. Know the large effective variables that are influencing your measurement of interest. Really huge effects on your ability to say, is this significant, is this not? You don't just want to average out over things, you'll lose. And then it's important to know the assumptions that your tests are making and to validate them. So that's just my little speech on good statistics. So, and then a short plug, this is the company that I work for, Koch Biological Solutions. They acquired Mendel Biotechnology, which became Mendel Biological Solutions, and now we're turning into Koch Biological Solutions. We are there now. It's been a DBA, and we're looking for biological products that improve crop productivity, and it's a super fabulous team of scientists that I really enjoy working with. So really recommend them a lot. And if you want to know more, you can go to these websites. So thank you.
Questions?
Yes.
Not a question, but one thing that will help you if you can express the residuals as normalized or studentized residuals.
Okay.
Gives you, see right now here you don't know how many standard deviations you're away from zero.
Yes.
If you normalize the residuals to unit variance, then you can look at numbers. You have 1 through 3, 4, and you say most of your residuals should be plus or minus 2.
Yeah.
Because based on the normal distribution, 95% of your residuals should fall in that range.
Yes.
And that makes judging them a lot easier.
Yes.
The other thing, a really neat tool, and you can do it in R, is to create a heat map of your residuals. A heat map is basically a two-dimensional map that represents a response variable as a color change. Mm-hmm. And had he done that, for example, in the previous talk with his row and column, he would have seen a very nice pattern. That's one of the more powerful tools there is. But overall, I enjoyed your talk and dovetailed nicely with what I had to say. You've got to look at your residuals. And no matter what test you do, I don't care if it's a t, chi-square, they're always assumptions. And your test stands and falls with those assumptions.
Yeah, and thank you for pointing out the heat map. I forgot to put that. That's a great idea. And sometimes you can see whether your outliers are lying on the edges of your field or whether they're just like spotty throughout your field, which can be a huge difference on whether you trust your ability to
Let me just do
throw them out.
One follow-up. If your company, a lot of companies are beginning to use JUMP now as a tool, and if you use JUMP, it's very easy in Graph Builder to make those heat maps.
Oh, okay. I've been writing my own code.
You don't have. But if your company uses JUMP, it's very easy.
I did not know that. Thank you very much. Awesome. Okay.
Yeah.
Oh, sorry.
And I don't work for SAS.
Yeah, thanks, Rachel. I fully agree with Ed. I mean, when we do all the field analysis, trial analysis, the first slide is a field map, and second slide follow this is heat map.
Yeah.
Heat map, is really good for you to see. I mean, it's actual yield, but the heat map, of course, with the residual analysis, I mean, it gives you more detail. And, you can see any residual, any pattern there.
Yes.
If you have a descendable pattern, as you said, we will do more. Yeah.
Yeah. Thank you.
Thanks.